Hi everyone. In the last video, we looked at how information is transmitted across a synapse. In this video, we'll explore the specific example of the cholinergic synapse and how different kinds of neurotransmitters can have different effects at the synapse, and also consider how drugs may affect synaptic transmission. A cholinergic synapse is one that uses acetylcholine as its neurotransmitter. As we saw in the last video, the arrival of an action potential to the synaptic knob causes calcium ion channels to open, allowing calcium ions to diffuse in, which then causes vesicles to fuse with the presynaptic membrane. In a cholinergic synapse, acetylcholine is released into the synaptic cleft and diffuses across. The sodium ion channels on the postsynaptic membrane are ligand-gated. They contain a cholinergic receptor as part of their structure, and when acetylcholine binds, the channel opens. This leads to an influx of sodium ions and depolarization. In this case, acetylcholine is an excitatory neurotransmitter, as its binding leads to depolarization of the postsynaptic membrane, increasing the likelihood that an action potential will result. If acetylcholine remained in the synaptic cleft, it will continue to bind cholinergic receptors, triggering further action potentials. It's removed to prevent this continued response. The enzyme acetylcholine esterase breaks down acetylcholine by hydrolysis into acetate and choline. The choline is absorbed back into the presynaptic neuron in a process called reuptake, where it is used along with acetyl coenzyme A to produce more acetylcholine. In the absence of acetylcholine, the sodium ion channels on the postsynaptic membrane can close. The synapses we have discussed so far have been excitatory, in that binding of the neurotransmitter to specific receptor proteins depolarizes the postsynaptic membrane and may result in an action potential being initiated. Some neurotransmitters can have an inhibitory effect at synapses. Their binding to receptors on the postsynaptic membrane leads to hyperpolarization. For example, they may cause chloride ions to enter the postsynaptic neuron, taking the membrane potential further away from threshold. This makes an action potential less likely to be initiated. It's also worth noting that while acetylcholine acts as an excitatory neurotransmitter at cholinergic synapses within the central nervous system and at the neuromuscular junction, it has an inhibitory effect within the heart. For AQA's exam, you also need to be able to predict and explain the effects of drugs on a synapse. So while you don't need to remember the details of the effect of any specific drugs, it's a good idea to consider some of the mechanisms they may use. Stimulants are drugs that increase the likelihood of reaching threshold and generating action potentials. Some may have a similar tertiary structure to the neurotransmitter and so mimic its effects, while others may inhibit the breakdown or reuptake of the neurotransmitter, allowing it to stay in the synaptic cleft for longer. Depressants have the opposite effect. They decrease the likelihood of an action potential being generated in the postsynaptic neuron. They may do this by blocking receptors so the neurotransmitter can't bind, or they may inhibit calcium ion channels in the presynaptic neuron, preventing the neurotransmitter being released. In the next video, we'll explore another concept that allows control of communication at the synapse in summation before applying this to the stimulation of rod and cone receptors in the retina.